We're very excited tonight for our presenter. This is the highest number of registered participants that we've had in all of our CADASILs to date. So clearly there's a lot of interest and excitement for this presentation, and we are very grateful. Dr. Arboleda-Velasquez is here with us this evening to talk about future for CADASIL therapy, overcoming challenges and bringing solutions to patients. Dr. Arboleda-Velasquez is an associate professor at Harvard Medical School and also the principal investigator at the Schepens Eye Research Institute of Mass Eye and Ear. He is trained as both a physician at the University of Antioquia in Colombia, as well as a research scientist at Harvard University. So the expertise is overwhelming. His work is distinguished by focusing on understanding the pathobiology of small vessel disease, including CADASIL, and uncovering mechanisms of resilience to neurodegenerative diseases, particularly Alzheimer's disease. But by bridging human genetics, vascular biology, and therapeutic innovation, his work aims to redefine how we understand and eventually present neurodegenerative and microvascular disease. Through collaboration across Colombia and the United States, his research is shaping the future of personalized medicine for both the brain and the eye. A very big welcome and thank you for being here, Dr. Arboleda-Velasquez.
Thank you so much, Elizabeth, for the very nice introduction, and to everyone for attending this webinar. I really I'm very excited about this consortium and everything that you're doing for the CADASIL community. So the title of my talk, Who Wants a CADASIL Therapy? Overcoming Challenges to Bring Solutions to Patients. I think who wants a CADASIL therapy? Apparently a lot of people, because many of you showed up today and many others are gonna watch this recording. So this is very exciting to see. I don't wanna forget my acknowledgements, because this has been a collaborative work done over many years. I want to acknowledge people in my lab, Dr. Said Arevalo, Paula Perez, Laura Correa, Annie Hewitt, Anisha Dosen, Tim van der List, and then Dr. Rafael Posada was in my lab, but now he's back to Colombia, where he has his own laboratory. Many collaborators from different institutions in Europe, in Colombia. I also want to acknowledge funding. So their work that I'm presenting today was funded by the NIH, and also some of that by the Department of Defense. And more recently, we had a very strong support from an organization that you may know, Think CADASIL. So very grateful for the support that allows us to produce the data that you're going to be seeing today. I was asked to present this disclaimer. This webinar is for information only, and the views expressed here do not necessarily reflect the CADASIL consumption, collaborators, or partners. It is not intended to provide medical or other professional advice. It is not intended, and also, well, I think that's repetition. Yeah, that's the disclaimer. I hope everybody understands. I'm speaking on my behalf. I'm also speaking on from my academic post. Other disclosures that are important, I am, some of the work that you're going to be seeing here and I'm going to be talking about today relates to a therapy based on inhibiting a protein called RANX1. So for us to be able to develop a therapy, you have to have intellectual property, and my institution filed a patent that has been issued for the use of RANX1 inhibitors, specifically for CADASIL. So it's important that you know that. And also, not only we have to make discoveries, sometimes we academics also have to get involved with entrepreneurs to develop those therapies and bring them to patients. So I have to disclose that I'm a co-founder of X1 Biotech, a company that is focused on advancing RANX1 therapeutics towards helping patients with CADASIL. Now, a lot of the work that I'm going to be telling you about today, some of the work is unpublished. But I don't want you to like get the idea that this is somehow new or unproven. The work builds upon previous work done in my laboratory and the laboratory of others. These are papers from my lab. All of these, each of these, 1 to 7, are papers, academic articles that have been published from my laboratory that support this protein RANX1 as a therapeutic. And we were the first ones. I'm going to show you a graphic about that really started this work on RANX1. But then many other laboratories, this is what we call independent validation. Many other people got excited about potentially using RANX1 for vascular diseases. And they publish their own papers, and I'm not involved with any of them. So this is what we call independent validation. And just to give you here a visual of how it has worked, RANX1 is our therapeutic target. In my lab, we were the first ones to publish of the potential use for vascular diseases. That was in 2017. There's usually a gap, because we put everything we had back then in that paper, but then we started to publish some more in different vascular diseases, different models, and we publish more papers. Each of these dots represents an article. And then, of course, people start to publish their own papers. So the work that is being presented today has a very strong foundation. Now, let me tell you something about RANX1, because you are a CADASIL crowd, so you usually hear only about Notch3, right? But there are other genes out there, and there are other proteins out there. One of them is RANX1, it stands for RAN-related transcription factor 1. And believe it or not, they have something in common. Notch3 is also a transcription factor. It's just that it happens to be in the plasma membrane or in the surface of the cell. But it has that commonality. So RANX1 is a transcription factor. And what I want you to know about transcription factors is that they really operate as nodes that regulate cell signaling. A lot of signals from outside the cells are growth factor, hormones, and stress signals. Usually what they do is that they regulate transcription factors. They regulate the expression of other genes. And RANX1 is one of them. And RANX1 is a very important transcription factor and is critical for differentiation of cells, for survival of cells, and proliferation. And some of that work was done by others many years ago. What we have done, and people were interested in RANX1 because of its role in leukemias, but we found out that it was also very important for vascular diseases and for fibrotic conditions. And that has led to our work now on CADASIL. So this specific link between RANX1 and CADASIL is rather new. I think in my laboratory we started to work on this maybe 2023, 2024. But it is now well established by different methods. When you want to establish a link between two things that appear unrelated, it's like, well, CADASIL is about Notch3. Why are we talking about RANX1? Is this link real? Well, we have evidence from human tissue. People that have died with CADASIL, they have donated their brains, and we studied those brains, and we found that this protein seems to be upregulated. We find a lot more of it in the brain of CADASIL people. And that's evidence that may be playing a role. Also, we have generated mouse models of CADASIL. So these mouse models have mutations in Notch3, similar to the ones that we find in people. And when we look at the brains and the eyes of this animal, the eyes also have like arteries. We find that RANX1 is also upregulated. We find a lot more RANX1 in CADASIL models. So now you see is in human tissue with CADASIL, in mouse tissue with CADASIL, And also we have found other, so this is very direct evidence, I will call it, but also we have found indirect evidence. Indirect evidence means in other models that are similar. Like for instance, people with diabetes, they have problems in their arteries, which in some way looks like CADASIL. And guess what? In those, we also found RANX1 as being kind of like upregulated. There is more of it. So that means we have direct and indirect evidence that support the importance of this mechanism. Now, I have been talking a lot about like problems related to the arteries or what we call the blood vessels. CADASIL is, or what we observe when we look at CADASIL brain, is vascular pathology. And in general, this is a transversal section of a vessel. It's like a tube, and if you cut it transversally, it looks like this, like a circle. So here is where the blood is, and this is the wall. And this is in a controlled brain, but in CADASIL, the cells that should be covering that wall, the small muscle cells or pericytes, are gone. So the cells degenerate and die. That's really the way we understood the disease. And instead of those cells that should be in these arteries, what we find are these deposits that I'm sure you heard about, the granular eosinophilic deposits that seem to be associated with the disease and are critical for diagnosis. For like, do we have CADASIL or not? You can do a genetic test, but you can look at these deposits in the skin. And if they are present, that's diagnostic of CADASIL. Now, there is an interesting correlation here. As you know, CADASIL is caused by mutations in NOTCH3, and the cells that they generate, in this case, the muscle cells and the pericytes, are the ones that mostly express NOTCH3 in the adult tissue. So there is a very nice correlation there. As you know, there are many of you are here because you are patients, so you or someone you know has a NOTCH3 variant, and they usually involve like letters like a C, a number, 170, and then it goes to another amino acid or another letter that is like an R or an S. That is a mutation. It should be a C in there or the other way around. So these mutations are very characteristic, and they happen in NOTCH3, specifically in this region of the receptor, and there are many different types of mutations that have been discovered all over the world. But they all have kind of like, most of them have that characteristic of involving cysteine residues. Cysteine is a type of amino acid that likes to make bonds. And in that way, it's very promiscuous. It can make bonds with like another cysteine amino acids if they're nearby. But if it's not paired, it will find another one in another protein and form these clumps of proteins that people think are toxic. And actually, some of these granular eosinophilic deposits seems like they contain NOTCH3 that is kind of like clumped up. And that is important for the disease. Now, how do we get from this type of like, we call it pathophysiology. Okay, there's a mutation in a gene. How do we get now to pathology? I think there has been a lot of research in the field on that and trying to figure out what we call the mechanism. But studying mechanism sometimes is different than finding a treatment. And I think we have devoted now, and I've been studying CADASIL since 1998. When I was back in Colombia, and I feel that's great. I think I gotten a good understanding about the disease, not complete, but enough for me to not really be focused more on therapeutics. So for me, for the mechanism, I would say enough already. We need therapies, and we need therapies now. Then we can do that in parallel with like better understanding. Now, there are other types of mutations that I want to bring to your attention. There is this beautiful paper that just came out like end of last year that shows us that, yes, in CADASIL, we have specific mutations that involve cysteine residues, or these cysteine amino acids that interact with one another. And that leads to what we call CADASIL. But there are other people that also have genetic variants in NOSH3, and they have a disease that affects the brain. That's why we call it cerebral. It's inherited in the same way. It affects the subcortical region. It even causes the leukoencephalopathy, those MRI kind of like spots, that the white spots that you see in the MRI. But some researchers just don't like to call it CADASIL because we are scientists, so we like to classify things into groups, and people can get very upset about this type of classifications. So if it's a cysteine variant, you can call it CADASIL. If it's not a cysteine variant, but you still kind of like get some pathology in the brain that looks like it, then those mutations are different, and they are very classic loss of function mutations. So in those variants, you don't have problems with cysteines, and you don't have these clumps or gums. But the pathology in the brain is very similar. Now, if you ask a patient, should I care about that? I would say it's important for some aspects of genetic counseling. Some people have found that these mutations that are like loss of function have like less pathology, but you can still get a stroke, and the MRI still looks like you have pathology. Instead, if you have the cysteine mutations, those tend to be associated with like a more severe phenotype. Now, we need therapy for everybody that has a mutation in NOSH3. The classification are for us scientists to like really try to distinguish things. And then a genetic counselor can make an assessment about which specific variant you have and how that will impact you and your family. But I think this article is quite interesting. And again, as we think of therapies, we have to think of therapies for everyone. Now, one of the goals of the talk was talk about barriers towards finding therapies. And I think it's fair to say that this has been a barrier, kind of like how so much we focus sometimes in particular mechanisms instead of focusing more on ways to treat the disease. What if we find a therapy that can help people that have all types of mutations in NOSH3? Wouldn't that be awesome? Wouldn't that be great? But that means we have to break a barrier, this barrier of classification. Now, I'm going to show you some evidence here about what we observe in the CADASIL brains. And for that, I want you to kind of like learn a little bit about the structure of the brain. And here is a representation of, I already told you about the artery that looks like a tube. It has the vessel wall. Here it has the endothelial cells. Here it has some small muscle cells and pericytes. Outside the arteries, the job of the artery is to feed the brain. And the brain is composed of, you can call it like two types of cells. One will be the neurons, that are the ones that do the thinking, and the other ones are the glia, that are support cells. And some of those glia are called microglia, that are very important for the immune reaction. So we call this thing, or this structure, the neurovascular unit. And it has the neurons, or let's say the microglia or the astrocytes. Then it has the endothelial cell and the pericytes. Well, what we did was that we got this human brain tissue, and we did a global analysis that looked at the expression of all the genes that they had. We can get that brain tissue from patients, and then we can analyze. And we had some people that had CADASIL and some people that didn't. And we do an expression profiling, looking at the expression of every gene. And when we compare the CADASIL versus the control, we found some things that were expected. Look at this. We found notch signaling, right? It's like, well, CADASIL people have mutations in Notch3, so no surprises there. But it's good to see that Notch was modulated because it may be told that the analysis was done correctly. And then we found two categories that are important and a little bit more surprising. One is angiogenesis. Angiogenesis is this phenomenon where you can make new vessels. Now here we are not showing that the brain is making new vessels. What we're showing is that there are some genes important for the formation of new vessels that seem to be regulated. And each of these red dots means kind of like increased expression or regulation, okay? And we also found inflammation. Inflammation happens also in the brain, like it happens in other organs. So as you can see, there's a lot of words, but I'm sure you can agree there's a lot of red here and less red here, right? That is, that means inflammation is increased. And the same thing here for like angiogenesis, I would say, and then kind of like also here some changes for like notch signaling. So there are some changes here that gave us an idea that there was a problem with the vessel related to angiogenesis. That is somewhat counterintuitive for the following reason. Remember, I was telling you how the vascular cells were like gone, that the smooth muscle cells or the pericytes were gone. And here we're saying that there is actually new vessels. Well, sometimes when you have degeneration, when you have cell death, you have a reaction of the tissue to actually produce more. And is new information here. CADASIL has never really been characterized or defined as an inflammatory arteriopathy, but here we're finding evidence of inflammation. And we not only can look at the gene expression, that's looking at RNA, we can also look at the protein expression. And here we look at it as a brown staining. So this is the slice of a human brain in a control, and brown is the expression of RANX1, and there is very little brown. Instead, I hope you can agree that there is a lot more brown in CADASIL brain. Now, what was interesting was that this expression was not only in the vessel, it was also in a cell called the microglia. Remember, the microglia was the cell in the parenchymal brain that is there for support and it mediates inflammation. So that is a very important finding. And here these charts telling us about like, kind of like quantify the result, and the stars indicate that they are statistically significant. So there is definitely an increase in RANX1 expression in the microglial CADASIL brains. And we have found that we found that in people that have different CADASIL mutations. So that seems to be a theme. There are many ways. Now, when you are doing research, I think the research I usually do is not very complex. It's very simple. Like I like to do simple research. Kind of like, is RANX1 important for CADASIL? If it is, well, maybe RANX1 will be really abundant in CADASIL. Now, I don't want to look at many different points. I want to show that one point in many different ways. So here, I'm showing you protein expression using a staining, and it seems like it goes up. Here, I'm showing you kind of like the same result, but more specifically looking at RANX1. So if I'm correct, I have to be able to find it in this list. You guys help me out, okay? So a big circle means big expression, okay? So let's go, and this is the list of genes that we found are kind of like regulating CADASIL. Let's see if RANX1 is here or, oh, there it is. And it's a big circle, and it's in the microglia, okay? So we found that the gene expression is there, the RNA expression is there. We found here we can run those, not just RNA, but we can also run proteins on a gel. And we indeed found RANX1 expression, these bands here, and the intensity of the bands indicate whether or not you have RANX1. And we see here light bands, and we see here stronger bands. And we can actually do calculations, and again, the stars indicate the statistical significance. I mean, we can look at it in many ways. I think I did it in so many brains using so many different methods that I convinced myself that it's true, that RANX1 indeed goes up. And we found that is indeed present in the microglia. So again, we want to do very simple, clear-cut research. And it seems that we have evidence that CADASIL goes up in CADASIL brains. Now, from human. Now, we make mouse models, and this cross-species validation, I mean, we are very similar, but at the same time, we're very different than mice. So we can put the CADASIL mutations in mice and see if we find the same result. Sometimes you cannot replicate what you find in the human in a mouse. But when you can, that gives you a lot of confidence in your result, because that means that your result cross species, okay? And here, what I'm showing you is some data that in animals that have NOTCH3 mutations, in this case, I show the data from a NOTCH3 knockout animal, meaning an animal that doesn't have NOTCH3. Why is this important? Because there are people out there that have NOTCH3 mutations that look like this, and they get cerebrovascular disease. And in analyzing these cells and tissues from the mice, we found the same result. We found here, these are the levels of RANX1 expression. They go up, and again, the stars, that means it's statistically significant. And here, RANX1 expression is in the tissue and is in green. Like here in the normal mouse tissue, there is less green, and here there is more. That means it's up. So again, RANX1 goes up in human brain tissue. RANX1 goes up in a mouse that has a NOTCH3 mutation. This is no CADASIL specific mutation, but it's a mouse that has a NOTCH3 mutation. And then that, to me, was enough evidence to go ahead and try to identify a potential therapy. So again, if RANX1 goes up and that somehow causes pathology, what if I can bring it down? Again, very simple research. Sometimes researchers think that they are so smart. Not really. I mean, we can make it very complicated or very simple. In this case, this is very simple. RANX1 seems to be going up, and I want to bring it down. And there is a drug called RO247429. It is a small molecule. This is, what do I mean by a small molecule? It's something that you can put in a pill and take it, like at home, as if it was an Advil, okay? So this drug, why does it have a weird name and not, like, why is it not called like something like Advil? Well, it's because it didn't get approved by the FDA. The drug was developed for a different indication in the 1990s, and it was tested in people with HIV at the time of the AIDS epidemic. And they, it made a phase 1, phase 2 clinical trial, did not advance because the patients were not getting better from their HIV, but the drug was found to be safe. So the patients were not harmed, but they didn't get better, and eventually our drugs worked better and they got approved. And that was what happened in the 1990s. Then a group of researchers at the NIH, again, not my work, at the NIH, a group of researchers was looking for RANX1 inhibitors, and they found out that RO247429 was a RANX1 inhibitor, and they published that work. That was done directly at the National Institutes of Health, not my work. So this drug exists and has been around for a long time, and someone else at the NIH published, independent of my work, that this drug is a RANX1 inhibitor. So what have we done here? This precedent gives us a great opportunity for what we call a drug repurposing exercise. When you do drug development, sometimes you first have to identify the target. And let's say I identify that RANX1 is a great target for CADASIL, and I want to find an inhibitor. Well, I have to do a screening of thousands, maybe sometimes millions of molecules. Then I have to do preclinical work. Preclinical work is I have to put in cells, and then I have to put in like mice, rabbits. Then I have to do sometimes monkeys. I have to do clinical trials, then do approval, and so, that costs hundreds of millions of dollars, and it can take 12 to 15 years. But there is an alternate path, that is the repurpose path. And repurposing drugs is great for a rare disease like CADASIL. In the repurpose path, a lot of the work is already done. The toxicology was done. The in vitro tests were done. I'm going to show you some mouse data that I did with this drug. So what you can do is do preclinical, meaning you show that it's efficacious for, let's say, CADASIL. You can go right away to clinical trials, and you can skip a lot of steps and save a lot of time. So a repurpose path can be done in 3 to 5 years if resources are available. That's why that was one of the reasons I got excited about this molecule, because it can shorten the path. I've been working on this. I think I've been honest about this with the community for like almost 30 years now. So I feel like I'm late, and I feel like I have not delivered. I want to deliver to the CADASIL patients, and I think this offers me of opportunity of a shortcut. I did spend a lot of time thinking about mechanism. Okay, I admit it. But now I'm taking a shortcut to see if I can deliver with what I want. It's my life now. I'm 50 now, and I want to deliver on this promise I made to the CASI community of a therapy. And maybe this is a path, but I cannot start here. I think I'll be too old by the time I'm done, and I care about the patients now. So I'm taking this repurpose path. And this is some data. The work is available. That's another good thing about this drug. Is published. The toxicology is publicly available. There's nothing secret here because it's all data. And the more I dig, the more I find. So that's fantastic. That's different than when you are making a drug and everything is secret. I don't want to belabor the point. I think the papers are there. The drug was published. It was tested in people, and it was tested, not a few. 6 people took this drug for 12 weeks. At 3 different doses orally. That's a big thing, you know. And they, you know, they it didn't work. I told you they still got HIV and AIDS, you know, but it was safe. Now, when what is to be safe? All drugs have side effects, but the problem when you start with a non-repurposed drug is that you don't know the side effects. You have to do clinical trials. Sometimes you find some things in animals, but then if you do, you cannot get to humans. But a lot of the side effects happen in humans because that's when you're in the clinic. So you only find out when you're in the clinic for the first time. Here we know actually, and we know what the side effect is. Some people got like a skin rash. Okay, so I want to tell you about it because the information is available. Some people got this skin rash. You know, these are the number of rashes, the week. But as you can see, if you're gonna get it, you get it early, and if they remove the drug, they went away. So this was considered to be a very mild. And if you, so I was like, well, you know, and I asked because one thing is what the drug developer thinks, and the other one is like, what do the CADASIL patients think? So I asked some people from the community, what do you think of these skin rashes? Is it a big deal? It's not a big deal. What do you think? And what I heard from the few people I talked to in the community was like they can live with it. If they get it, they can live with it. Or another one that I heard is a stroke is worse, or migraine is worse. Another one that I heard was like, I'll wear a sweater. But again, we'll have to advance, use use the drug, get a trial and see what happens. But it's also dose dependent. So we're seeing more with the higher doses. I don't know if we're gonna need the high dose here, but because it was 75, 150, and 300 milligrams per day of the drug. So the rashes were more obvious here with the 300, okay? But maybe we don't need that much of it. What else I wanted to tell you? This drug now is commercially available simply because the drug was dead and we resurrected the drug. So we paid the company somewhere during the pandemic for another indication to bring the drug back to life. And because we published, people started ordering the drug. So this drug is now available from commercial sources for research purposes only. But why am I bringing this up? Because I definitely don't want you to go out and buy this and take it. Do not do that. I don't think it's safe. Why is it not safe? Because this was made for studies in cells and in animals. It's not human grade. Okay? Um, I hope that is very clear. But the advantage of the drug being commercially available is that everyone in the community can go out and buy and do experiments with it. So you cannot do that a lot of the times when the drug is in development and is not repurposed. Here you can. There are some researchers here. And we want to publish the work and everything. People can go out and validate themselves in their own models if it works. So this is fantastic. They can buy from Medcube. They can buy from Fisher. There are many providers. And again, it's available because my lab resurrected this drug. And now people can play with it, okay? A lot cheaper. I had to pay tens of thousands of dollars the first time, but now people can pay for $300, okay? So what we did is we took the drug and we wanted to do a very simple experiment. What we had was an observation. The observation was simple. The observation was RANX1 goes up. It goes up in human brain of people with CADASIL, and it goes up in mice that have a notch mutation. Actually, we also tested that in a CADASIL model, two of them actually, but here I show you only the knockout data, and RANX1 goes up. But that could be simply like an association. What if maybe many things go up and many other things go down in CADASIL? Who cares? What we had to do was kind of like an experiment where we put drug and we see if it has a beneficial effect in the model. So in this case, we use this model. It's a typical CADASIL mutation. I told you, a C, that's the system, followed by a number, followed by another letter, R. Pretty typical CADASIL mutation, and we have tested now this in two different mutations. We also tested a mouse model, the arginine 1031 system mutation. What we did is that we took animals that were already pretty sick. They were 6 months old. At that stage, the animals get severe pathology, and then we treated them every other day for a month. So they were 7 months by the time we analyzed them. And then we look at their retina, we look at their brain, we did some analysis. And what we found was remarkable. This is, we're looking at their eyes. I know there are some efforts to also look at the eyes of CADASIL people, but I joined the Eye Institute a long time ago. So we do eye research and we look at the eyes of the mice. And what I'm showing you here is a live mouse that has a CADASIL mutation, and then we can stain the arteries with the dye, and it's green. So what you can see here are the arteries, and actually, I can tell you what's what. This is a vein. This is an artery. This is an artery. So if they are really wide, they are like veins. So in CADASIL, I'm sure you can appreciate these vessels are really kind of like dilated. And that's because they lose the smooth muscle cells. So the vessels kind of like are really fluffy. And that leads to like leakage, microhemorrhages. It's ultimately related to stroke. Okay. So in the CADASIL mutant, they are really kind of like dilated. But then when we treated them for only a month, the change was quite remarkable because we can see that the vessels became like very thin again. And I thought that was, and we quantified that, and it was significant. And I thought that was kind of like really unexpected, that we will see such a robust effect that I could tell. We have done this exercise, like they spread all the images and to see if I can sort them, and I can't. And I get everyone right. And they have also tried to do that with AI, but we can sort which ones. I can sort which ones have a CADASIL, which ones are treated, which ones are untreated. There's no scientific; it's just satisfying that I can do it. But they can do also some quantifications, as you can see here. And then, but it was hard to explain because remember, the ones that maintain kind of like the structure of the vessel are these small muscle cells. So that kind of trying to explain, and again, we go back to mechanism, trying to explain how this is working and why it's working, it's probably going to take us a while. But let me tell you what we observed. What we observed was here in red, that's a normal mouse artery covered in the small muscle cells. And all of this red means all the cells are where they are supposed to be. In the CADASIL mouse, those cells are diminished. And in the treated mouse, those cells are there. Okay. Now, this is a little bit puzzling because, we treated for a short period of time. And also because the understanding in the field was that in CADASIL, the cells were just dead. But so how can you bring them back somehow? But people have been changing their understanding about vascular pathology in conditions, like for instance, in similar conditions like in diabetic retinopathy. We also thought that the small muscle cells and the pericytes were dead, and they weren't. They had just left the vessel. And the moment you do some specific treatments, they come back. And I do wonder if this is a part of what we are observing here, that there is this recovery of the vessel. And again, this is unprecedented. I've been doing this for a long time. This was not what I was expecting. I have done experiments differently, which is preventing the loss of. Like those cells are going to die, and I start treating the animals so early that they don't die. So they are protected from death, not like back from the death. Okay. But I didn't do that experiment here. I did the experiment. I'm really thinking about people that already have CADASIL. So I did this in animals that are already very sick and have pathology. And the result is quite surprising. And it looks real to me because, this doesn't look like that. And some of these cells, actually, we see them sometimes like landing. They are like onto the vessel, but haven't landed yet. So we have like spectacular images with that. But here, just to give you an example, or like this vessel is so dilated that to be so dilated is because it didn't have as much muscle cells before. So now they are back, but the vessel is still dilated. Again, the result is quantifiable, and we know in CADASIL diminish small muscle cell intensity, and here it increase. Which is quite a remarkable result. But this happened in mice with the C455. It happened with mice with the arginine 1031, two different calcium mutations. And I don't think we did it for the knockout, but we did we did the retina for the knockout, and it was also recovered. Now, we also look at the brain, and in the brain we found a similar result. Now, what we think is happening here, and I know there are some scientists and some researchers that might be interested in this, what we think is happening here is that the RANX one, I mean, where does the pathology start? It probably starts in the vessel, because it's a vasculopathy, right? So there is some vascular cell, the vessel starts to leak. Then what happens here is, this is a microglia. The microglia are usually like close to the vessel, but not on top of it. What happens in CADASIL is that really the microglia goes to the affected vessel, and it's kind of like trying to patch it up. Like you can see here, the vessel is in like blue. So it's really kind of like trying to like patch it up. And in that process, the microglia gets activated. And we think that that kind of like starts a vicious cycle where the microglia make it worse. And they start expressing RANX1. So what we think is happening is that the moment that we put the inhibitor, we block the vicious cycle. Am I doing something to NOTCH3? No. Am I correcting the genetic defect? No. But we are helping the pathology so that the vessel has a better structure. And this is what the drug is achieving. So I think RAN inhibition, RANX1 inhibition, we can change this. I mean, it reduces it restores vascular reactivity. Those are the kind of like the fluorescent angiographies, the imaging of the retinas. It reduces the vascular smooth muscle cell loss or restores the coverage. We have to think of some proper language to call that, but I think the remarkable result is there and it's quite reproducible. And also RANX1 may be a treatment for CADASIL. That is what I pose to the community. We have significant evidence. I want to leave some time for questions, so I'm just gonna say that these drugs have some pros and cons. I'm excited about it. A lot of other people are excited about it. We're able to convince a company of trying to advance it. I talk about that. We have evidence that it works in models with different CADASIL mutations. The drug has been tested in the clinic and is oral. I think this is a huge advantage over other drugs that you might have heard about for CADASIL, which I think would require like infusions or even spinal taps for that. Now, repurposed drugs are difficult to fundraise for because people are like, well, that path is too short, and yeah, you can help patients, but I'm not gonna make as much money as I would otherwise, so I'm not gonna invest in the effort. That is there. Anyway, what I started recently was trying to socialize this discovery with the community. Doing this seminar is part of that, but there is more. I was invited by Carrie Pasquarello. She's an entrepreneur and she has her own podcast, and I agreed to do my first podcast. So we're gonna record it this week, and then you guys can watch it on YouTube, and hopefully it will be good because it will be more conversational. Now, with that, I wanna end my presentation. I think, yeah, we're gonna record it this week, and then she's gonna like broadcast it the week after. So many thanks to Carrie again, and many thanks to Think CADASIL for all their support, and they have provided resources for the research that you're talking here, that I presented here. Thank you. Now I open up for questions.
Thank you. Thank you. The date of that podcast or with me, I can put that in along with the recording that I send out to folks and
All right.
give them an opportunity to log in for that. That sounds wonderful. We do have several questions, and I know I promised you would be able to pick your son up on time. So I'm going to, for the sake of time, try to combine some of the questions. We've got 6 on the docket. Most of them you did touch on during your presentation. So if you need to refer back or you want to just refer that way, we can do that. But I'd like to try to combine some of these so that at least we touch on all of them. And the two I can combine first relate to treatment. One question reads, I understand that this medicine is being looked at for possible treatment to slow or reverse the progression of the disease. How can we get info on clinical trials? And then as a related question, what are the barriers to clinical trials right now? Is the thinking that this will be a course of treatment or a pill that would be taken for life?
Okay, so these are excellent questions. What I will say is, will it barriers? Well, where do we stand with this drug? Yes, it was in the clinic, but it was also kind of like studied like a long time ago. So my understanding is that the FDA, and it already passed all the toxicology tests. My understanding is the FDA wants us to repeat some of that toxicology study that were already done so that they are done under current standards. That would take some time and resources. And once that is done, we can launch a trial. How soon could that happen? I mean, we're still in April. If resources are available and we do talks this year, we could and everything is clear, we could theoretically start a clinical trial next year. Because again, that's the point of now, is the clinical trial available right now? No. Some people have asked me, can I enroll? No, you cannot. What you can do is participate of these studies where, let's say this works. Let's say the resources are there, we do the talk studies, they come out fine, and we can do a trial. We are gonna enroll, call Elizabeth and Jane Paulsen and see if we can enroll people on the trial. We're gonna call the consortium and other clinicians that have patients and see who wants to do it. So you guys are already doing the work. You're not missing out because we will use the biomarkers that are being studied by the consortium for instance, hopefully. This is all theoretical. We haven't really talked about that specifically, but we will look for patients here in Colombia and in Europe and work with whoever wants to participate. Will this be something that you have to take for life, or you take it and you're done? It is chronic treatment. I just don't know if you have to take it every day of every year, because the effect was very strong. So I do wonder if we can do like a cycle of treatment for like a month, then you're fine, and then you do another follow-up, and in 6 months you do another cycle. That'd be fantastic. Not unheard of for when you treat transcription factors. A big example is steroids. Have you heard of the steroid cycle? You take it for like 5 days, right? And that's it. Then the effect is done and you are in the clear. Of course, I would love for the treatment to be like that, because even taking pills could be onerous. And what if the rash it bothers people? Well, then you do a cycle and then you can be without the drug. It can be it's great to know what the side effects are, because then you look for them and you work around them. I think I answered all the questions that were packed in there.
Yes. There was a follow-up asking if there were any clinical trials or research in Colombia or even other countries or places that you're aware of.
For this drug, no.
Okay.
Actually, that no. I mean, maybe somebody starts that, but again, no. No, I want to make it clear, there are no ongoing clinical trials for this drug. We want to do them, but they are not ongoing.
Okay, I'm going to try to combine two more questions here quickly, and they both relate to the first asks, is it RUNX1 cause Notch3 mutation, or does the mutation create more RUNX1? And in a related fashion, where can we find more information about the variants? This person's family has 505C. So just kind of asking more about that.
Okay, so in this case, we have tested just that it works with Notch3 mutations. The location or the variant is not going to matter. And let me tell you why. Because the effect is not direct. So that goes back to your question. That's NOSH. I think NOSH3 mutation ultimately causes RANX1 to go up. But the effect is indirect. And because it is indirect, it may work for any and all NOSH3 mutations. If it's direct, then it depends of which mutation. And we have had some treatments that are very specific for a specific position. So then you kind of have to be lucky to like be right on that spot. I don't think this applies to this drug. This drug works for people with diabetes that have vascular abnormalities, similar to CADASIL. So I think the effect is, they are linked. The link is indirect. And I will expect that it works for people that have any calcium, any of the positions. It should work. And I have tested two. Very different one. One is at C2R, the other one is an R2C. They are different. They both work.
Thank you. I've got just two more. I'm going to try to combine. There's a few more, but I think I can combine them to make it just down to two last questions. This is about whether anyone is using AI to find a cure for CADASIL, and the person who asks it suffers from tonic-clonic seizures. And so the follow-up question is, why do 10% of the people that have CADASIL also suffer seizures? And their family it also runs in their family. Brother and father had the same, and just hoping to see if AI or any other methods are being used to advance the possibility for a cure.
I think everybody's using AI. I'm using AI. I use AI to prepare this presentation too. I think it helps. I have used AI more directly. I'm not aware of particular efforts to do drug development, but there is a misconception about AI, which is that, let's say AI can design the best drug right now for CADASIL. I can tell you it's going to be new. So even if you find it right now, you're still 12 years off. The bottleneck for drug development is not discovery. That is actually, I mean, a lot of people will agree with that statement. It's not discovery, it's advancing the drugs and making sure they are safe and effective, which means dragging them, which is an actual dragging of the drug through preclinical and clinical development. Now, the seizures have to do a lot of the, I mean, when the vessels dilate and they leak, they cause a misregulation of, a lot of things that shouldn't be in the tissue leak out, and that manifests itself as a seizure. I will expect, which, like, so this leads me to, and I would like to end with this. What will I expect this therapy to do? This therapy will restore the vessels in a way that they will leak less. So the MRI, I will expect it to be cleaner. Those white spots should be reduced. And also I will expect a reduction of the microhemorrhages and microaneurysms. And yes, I will expect a reduction in the frequency of strokes or the recurrence of the strokes or the severity of the stroke. So what I would expect this drug to do is to help. Ultimately, that will improve cognition or kind of like stop the progression of the disease, which is one of the problems with CADASIL. It's not just that you have it, it gets worse with time. More limitations for mobility, more cognitive decline. What if you kind of like stop progressing? That would be like quite an ideal scenario, because then you can do other things and the brain plasticity will kick in and maybe you even see some recovery. I do not expect the drug to directly trigger recovery. This is not that type of drug, but it may stop progression such that you can then do other things that allow for actual recovery. And again, this is important to like, well, let's say this drug works, what should I expect to see? From a biomarkers perspective, a cleaner MRI, I would say that should be observable. I mean, I just show you like the image of the retina. It's very, it's very clean, right? And then from a clinical, but that will translate into limited progression of the disease, which I think for CADASIL that'll be a breakthrough, and I'm very excited about that possibility.
Thank you. Thank you. I did promise we would finish you on time or finish for you on time. So I think with that, I will also mention that next month's presentation is going to be May 27th, and that is going to discuss specifically MRI findings within the CADASIL research. So everything everyone is already doing to help expedite the research, volunteering, asking your family members to volunteer, being a part of this is really being a partner with us in finding solutions. And Dr. Arboleda-Velasquez, we just thank you so much for your presentation tonight. For the questions that were not able to be addressed, I will try to address those personally with folks and post where you can find more literature about this and other potential avenues for treatment. So thank you so much, everybody. I hope we can see you again next month.
Bye-bye. Thank you for the invitation.
Thank you.